Hi students, welcome to Year 11 Biology and Cells as the Basis of Life. This is video 4A effectively, and we're just going to continue on to look at the relationship between our understanding of cells and also the development of technology. So the same thing that we were doing before, we previously, on a previous video, looked at the light microscope in a little bit more detail. That's the most common one. It did allow us to look not only at different types of cells, but also to start looking at some of those structures, those organelles that we find inside the cell, and learning a little bit more about the cell by being able to see some of these additional structures. So in this video, just a couple of additional ones that you may come across. One of them is phase contrast, the phase contrast microscope, and this one looks very much like a light microscope and works similarly in some aspects, but primarily the difference is that it is trying to create a kind of shadow of the specimen. Here, for example, is a sample of E. coli. So you can see what we're trying to do here is contrast, in this case, the cells, the bacterial cells, the E. coli cells, against a gelatin background with something like an egg cell or oocyte. Again, you're not getting the sort of resolution that you get with a light microscope, but you are able to see some differences in different regions or cells against a different background. And the other one to talk about, which is very highly likely that you don't have in your school, is an electron microscope. These are expensive microscopes. They do not rely on light. They rely on a beam of electrons, not light. And this is great because it means that we can massively increase the resolution. Electron microscopes have allowed us to see structures way smaller than we could ever see with a light microscope. There's two main types of electron microscopes, scanning and the transmission electron microscopes. I don't know that you need a lot of information about each of these, and we'll probably look at them briefly in class just to fill a little bit of stuff out for you. But the key here is you're looking at light, which probably can give us maybe 1,000 to 1250, something like that, times magnification. That's about as high as it goes, and usually you're using something like oil immersion in order to get that kind of resolution. In our electron microscope, it's massively bigger. We can see much smaller structures, and in fact we've learnt a lot about the microstructures, the organelles that are present in cells from the development of the electron microscope. And as you can see, it's been around a little while now. It too has been developing, changing, being refined, improving the image, improving the process, and it's really allowing us to build a really good story around cells and the components that are within cells. To give you a little bit of an idea of the sort of stuff that you can resolve so much better with electron microscopes, a little pictorial, I guess, that gives you a look at the concave nature of red blood cells. So we know that they're cells that are very different from most of the other cells in our body in that they don't have a nucleus. They're really just little bags of hemoglobin transporting oxygen around. And you can see this shape is quite unusual. And if you contrast that with the image of the white blood cell that's present there too, which is this one, in comparison to these red ones, red blood cell and a white blood cell, you can certainly see some differences that are present there. Thanks for watching.